Hi folks, this week's lab content is diffusion and osmosis. So there's going to be some terms that we're going to need to put into place so that you understand what they mean, as far as obviously defining diffusion and osmosis, and a few things that you'll need to know to prepare for this week's lab. So one constant that we always want to think about is the term homeostasis. We can also look at a term called equilibrium and kind of look at them as being very similar in terms of what they are. Now, thus far, we've talked about measurement, scientific methods, so how do we do science, how do we study different things. We've also talked about how to use a microscope, and then what cells are and what they're made up of as far as their components. So now we are going to take that cellular level and we're going to look at what's happening in the cells pretty much for the rest of the semester. So we're not going to specifically look at cell structure, but we're going to talk about what's happening on the inside of the cells. What's going on with our different cells? And we're going to start with talking about diffusion and osmosis. So homeostasis within our cells is that the cells are maintaining a steady condition, an optimal condition for them to be that way. Because there's so many reactions happening inside our cells and even outside our cells to keep the cell alive, to keep the organism alive, there's lots of fluctuation. But the ultimate goal is to stay in homeostasis. And so that is done by kind of regulating what passes into and out of the cell via the plasma membrane. So let's take a look at the plasma membrane again. We looked at it last week with cells, but let's talk about it a little bit further again. So basically it's composed of what we call a phospholipid bilayer. So remember, bilayer means that there are two layers. So you can again see one layer here and the secondary layer here. So two layers, bilayer. Now what's making up this bilayer are what we call phospholipid molecules. And there are many molecules kind of stacked next to each other, and then you'll see it's almost like a sheet of phospholipids. Now, when we look at this individual molecule here, the circular part is basically what we call the head of the phospholipid, and the head consists primarily of a big phosphate molecule, but it also has a glycerol and a chlorine in there. So this is what we call hydrophilic, meaning water lover. And so when you look at this picture over here, these are all the heads on the outside of the cell, and these are all the heads on the inside of the cell, because as you can see, there's water on the inside and the outside of the cell. We know inside the cell, we have the cytosol, which makes up part of the cytoplasm, which is basically water-based with other substances in it. So water lovers are going to be facing the water themselves. However, if we go back to our individual molecule here, coming off of that phosphate, we have these little tails, which are basically the lipid part of the phospholipid. And we know lipids are basically fats. And so fats are not soluble in water. We know oil and water doesn't mix. So we call these guys, they have the tendency to be hydrophobic. They do not like water. And so they align themselves in the middle here. And so this is an area where there's no water, so there's no H2O. Whoops, no H2O. So this is where they kind of hang out. Now, because of that, a lot of substances and solutes are not going to be able to go through this as easily because of all these lipids in here. So we need to have different channels, for example, protein channels that molecules can go into and out of very easily. We also have channels that are pumps, so we have different ways for things to get across the membrane. So let's look at some of those important terms that we need to know about. And I'm going to add one here that I didn't type in, but we're going to add it. We're going to add what we call solution. So first of all, let's talk about a solute and a solvent. So a solute, basically an example I give here is salt, but it's basically any organic molecule, and the key is here that it dissolves in water. So if you think about your kitchen and your household, we might take salt, and dissolve it in water, but what other things might we dissolve in water? We might dissolve sugar, we might dissolve flour, cornstarch, a lot of our kind of powdered things in our cabinets, but we might also dissolve, for example, hot cocoa, iced tea, lemonade, and we make something new when we dissolve it in water. So when we mix it with water, the water is what we call the solvent. And in our particular lab, often, in many labs, water is the solvent. It's always the solvent. So when we mix a solute with a solvent, we get a solution. So a solution basically is a solute, whatever that might be, plus a solvent, water. Okay, and a lot of times what happens if we mix these together is they might stay liquid for sure. They might become like a semi kind of solid or have some form to it. So like for example, if we were to make Jello in your house, Jello is usually, whether it's, regardless of what it is, it's a powder form that actually has gelatin in it. And Jello likes to be mixed with a solvent such as water, and sometimes that water needs to be a warmer temperature for the gelatin to basically dissolve in it. But our solute is the gelatin in a powder form. When we mix it with warm solvent of a water, basically it becomes a liquid, right? It's a solution of the two mixed properties. Now, when we put that in a cold environment, for example, so when we make Jello, we put it in a dish or a pan or little cups, and we put it in a cold temperature. And what happens after a while is you pull it out of the temperature, so out of the refrigerator, and what is its form now? It's kind of a semi-solid. So we have that kind of gelatin semi-solid. It's not, if we mixed it right, going to be a liquid. If we, again, mixed the ratio correctly, it's not going to be a hard mass. So that is an example of mixing things. Now, equilibrium, like I said, there's no concentration gradient, and the key is that we are balanced. So thinking of like the seesaw, right, in the park. If this is flat, which I didn't really draw it correctly straight, we're at balance, we're equilibrium. However, if we have a kid that doesn't weigh very much and then another kid that's bigger down here, we are at a balance, right? And so we don't want that to be, because that's how our cells would be way out of whack if that's the case. We want to be straight. Oh my goodness, that's horrible. Balanced. So kinetic energy, we've talked about all things have potential energy, the ability to do things. So atoms and molecules are always constantly in motion. No, we can't see that, and that's that kind of kinetic energy. We can't necessarily see it because it's so small, but if we were in lab, we could simulate this, which would be really cool, but unfortunately we're not in lab at the moment. But let's talk about diffusion. Now, diffusion is movement of molecules from a high concentration to a low concentration. So their concentration is too much, like they're crushed together, too many of these little green solutes together, and they want to get away from each other, so they move to a lower concentration where there is less. So here this blue is kind of a solvent, water, our solute to these tiny little molecules, and they're all really close packed together, and they just want to move away from each other. They're like, I don't want to be near you, so I'm going to move over here. And this does not require any type of energy at all. This is just what we call passive. It just happens. So for example, if you were in an elevator with a bunch of people, let's say there were 30 people or so in the elevator, you might feel really claustrophobic and like really close to people, and you might be like, oh, someone didn't put deodorant on, oh, that person has too much cologne, that person is eating something that doesn't smell good. So you're in this environment, and that's a high concentration. Now when the doors open on the elevator, we all sort of fill out, and we might do it very uniformly, but we are moving to a lower concentration. The area, the vestibule outside of the elevator usually has more space, so we're moving to that lower concentration. Osmosis is a specific type of diffusion. So same thing, concentration high moves to low. However, the difference here is what's moving. The solvent is moving. The water is moving in this scenario, not the solute, not the molecules. The water is moving across the membrane. So that's really important to realize. And so when we're doing our experiments this week with the egg, be aware of what you're using. So am I using water and other solutes? What's moving? So that will define whether you're looking at diffusion or osmosis in your experiments. Now, the last kind of term that we have when we talk about things inside versus outside the cell is the tonicity. Now, tonicity, there are three types. there is isotonic, hypertonic, and hypotonic. And so each one of them has a prefix in the beginning that tells us a little bit more about it. ISO, I-S-O, always is equal. So we are kind of, again, balanced. This is the optimal. This is what we want to have, right? We want to have equal inside versus outside. And what we're talking about is just that. Equal solute and solvent inside the cell, outside the cell. Now, hypo, always a term that means lower or below. Now, what's lower, so what we're really worrying about is the change in solute to solvent. So in a hypotonic, we have a low solute, so a low solute of molecule, and therefore we have high water, almost like too much water, okay? Now, in a hypertonic situation, it's just the opposite. We have a high solute, okay? So we have a lot of molecules in that cell, and therefore we have a low water. And this is almost like being dehydrated. So the cells are kind of dehydrated. So let me show you a visual, because I think pictures are really important. Here on the left, we have our plant cell, and then on the right, we have what we call a red blood cell. So here's a plant cell. This is an animal cell, obviously. And I'm going to put RBC, which stands for red blood cells. So here is isotonic in our plant. So you can see equal water going in and out, and this is a typical looking cell. Here is our central vacuole in the middle. So it's going to hold on to that stored water and other molecules. But again, the water flow is going to be equal. Our tonicity is equal, so isotonic. Over here, if we're looking at isotonic of our red blood cells, so if you look at the red blood cells, isotonic, these cells are biconcave, which means that they kind of have this ability to go inward, right? So this is kind of what the cell looks like. It's almost like that shape. So that's normal, equal waters in and out. Hypotonic, so if we were to look at the hypo, which means we have low solute and then high solvent, which means that, like say for example, our solute is sodium. So our solute is Na. Okay, so here is hypotonic, right? So what happens is we remove the solute, so we have a low amount of solute, so we get bombarded with water, so water is rushing in, and so what happens is the cell actually looks round. It becomes a balloon shape in the red blood cells. Over here, what happens to the vacuole? It gets quite large, and what's actually happening is that it smushes all of the cytoplasmic material around the edges, and so things aren't going to work right in that particular cell. The shape's not going to change of a plant cell because of that rigid cell wall. So what happens is that the water vacuole fills up so much, pushes pressure, which we call turgor pressure, against the cell wall that it's going to kill the actual cell. Over here, if this becomes a balloon, what eventually happens, and we know balloons get too big, they pop, and that's exactly what's going to happen here. We don't have that rigid cell wall, so we have the ability for it to kind of explode, and that would obviously kill our cells. So again, low solute, high solvent, high water, too much water. So too much water in our cells can actually be harmful at times. The opposite of this is hypertonic, where here again we have too much solute and we have a low solvent, okay? So what happens then is, now look at our vacuole, it's shrunk. It's this tiny little thing. So it's almost like the example of being dehydrated, and everything, so this is all the cytoplasmic material, pulls away from the cell wall. It's only being held on by some of these fibers, but again, it's not going to function properly. And this is kind of the example of when your plants haven't been watered in a while and their leaves start to kind of wrinkle a little bit and they kind of curl up, and then eventually the whole plant might kind of lean over, is that you are in a hypertonic situation. You do not have enough water on board. You have too much of a solvent. In our cells, an example of hypertonic, again, here's our red blood cells. What happens is they start to shrivel up. So they're not kind of nice and plump and biconcave here. They're surely not big balloons. They're shriveled little tiny cells. And if they're shriveled up and they don't have enough water on board, they're clearly not able to do their job. And so therefore, they will still not function and most likely die if we're in that state. So water regulation is really important. And water regulation, the movement of water from high to low, is an example of osmosis. So when you're doing your experiments this week, make sure you are being aware of what's diffusion, what's osmosis, what tonicities do I might have, and where is things moving? Are they moving into the cell or are they moving out of the cell?